So good morning all of you. In this video lecture we will discuss about the all important events of cell division, that is mitosis. In the previous video I have discussed about your cell cycle, how the cell cycle occurs and what are the various steps in cell cycle. So let us now start mitosis. Mitosis was first coined, the term was first coined by W. Fleming in 1882. Well, mitosis is confined to somatic cells or vegetative cells. Okay, it only occurs in somatic or vegetative cells. It results in the formation of two daughter cells from a single parent cell, and the most important feature in these two daughter cells is that they contain the same number of chromosomes as the parent cell. Okay, if the parent cell is having 10 chromosomes, then the daughter cells, two daughter cells formed from that parent cell will also be having the 10 number of chromosomes each. Got it? So in case of plants, the mitosis is confined to the vegetative parts of the plant, which include your roots, stems and leaf. Got it? Whereas in case of animals, it occurs in bone marrow cells, liver cells and base of nails. Okay? As far as steps in mitosis is concerned, it is completed in two steps. One is karyokinesis and second one is your cytokinesis. Karyokinesis is nothing but division of nucleus. Okay? After the nucleus has been divided, then the cell enters into the cytokinesis phase. In cytokinesis, division of cytoplasm occurs. So both karyokinesis and cytokinesis, they combine together to form two daughter cells from a single parent cell. Got it? So ultimately we are getting two daughter cells from a single parent cell. Okay, and the chromosome number in case of two daughter cells will be having the same as the parent cell. Okay, now let us start the karyokinesis. Nuclear division, you know, karyokinesis is the nuclear division. So this nuclear division is completed in 5 steps. What are those? Prophase, PRO-METAPHASE, metaphase, ANAPHASE and TELOPHASE. Okay, prophase, PRO-METAPHASE, then metaphase, ANAPHASE and TELOPHASE. So we will discuss each step one by one. First come to the prophase stage. Remember, prophase is the longest. It is completed in the lengthiest time. It takes the lengthiest time. Initially in this phase, the chromosome appears thin and filamentous. Okay, but after dehydration and coiling, the chromosomes get shortened and thickened. That means in prophase, the chromosomes are very much shortened and thickened. Why it gets shortened and thickened? Because of dehydration and coiling. Got it? At this stage, the chromosome consists of two distinct chromatids connected by a centromere. Okay, so at the prophase chromosome consists of two chromatids. In case of animal cells, the centrosome is present and it gets duplicated during interphase stage and consists of two centrioles. Got it? You know it. Now these two centrioles move towards the opposite poles of the cell during the prophase stage. Hello? Okay. Now spindle fibers consisting of microtubules develop from centrioles and constitute aster. In case of animal cells, the spindle fibers consisting of microtubules develop from centrioles and constitute aster. Now central broader part is called equator and the narrow ends with aster is called poles. Okay, but as far as plant cells are concerned, centrosome is absent and spindle organization occurs in cytoplasm. Hence the mitosis is known as anastral type of mitosis. Okay, here asters are not formed, so this type of mitosis is called anastral type of mitosis. So let me draw. As far as prophase is concerned, this is the cell, this is the plant cell or cell wall. This is cell wall. Okay, this one is the nuclear membrane. There is presence of nuclear loss. Okay, Then the chromosomes, each chromosome consists of 2 chromatids. So let us take 4 chromosomes. 4 chromosomes in this particular case, rather 3 chromosomes, it will be easier for us. 1, then another chromosome, 2, 3. So 3 chromosomes are present. Got it? So this is what your prophase is. Now once the prophase is completed, it progresses and enters into the pro-metaphase. In some cases we call it as late prophase and early metaphase. Got it? So it is pro-metaphase. So what happens during this pro-metaphase? Here dissolution of nuclear membrane takes place. So nuclear membrane start to disintegrate. In addition to that, nuclear loss also disintegrates. Okay, nuclear membrane and nuclear loss, both of them disintegrate. Now there is due to disintegration of nuclear membrane, the distinction between cytoplasm and nucleoplasm is lost. Okay, as there is no boundary, so distinction between cytoplasm and the nucleoplasm is lost. Now the chromosomes are attached to spindle fibers by centromeres. Got it? Such a mitosis is called eumitosis. Actually, these spindle fibers are not directly attached to centromeres. Rather, some disc-shaped structures are connected to centromeres, and these spindle fibers are attached to those disc-shaped structures called kinetochores. Okay, this is the diagram of those disc-shaped structure. If this is the centromere, on its sides, these are the structures. Okay, these are the kinetochores. This is the kinetochore. To these kinetochores, your spindle fibers get attached. Okay, so this is what your pro-metaphase. So how pro-metaphasic chromosomes are arranged? Let us see. Let us draw another diagram. So this is the plant cell. So this is the cell wall. Now say nuclear membrane and nuclear loss, they are getting disintegrated. So by the end of the pro-metaphase, totally they get disintegrated. That's the reason why I have drawn it in a dotted line. Now nuclear loss is also disintegrating, so I have drawn it in a dotted line. So how the chromosomes are arranged? You know, each chromosome would be having 2 chromatids. So let us see. We have taken 3 chromatids. So in this way, one, another one, this one, this one. So these 3 chromosomes are attached. Got it? Now in addition to that, spindle fibers have formed and they are attached to the kinetochores. Okay, got it? So this is what this is the rough diagram of your pro-metaphasic stage. Now come to the next one, that is metaphase. What happens during this phase? Now the chromosomes which were migrating towards the equatorial plate. What is the equatorial plate? The equatorial plate is the middle portion of the cell. Got it? The equatorial plate is the middle portion of this cell. So now the chromosomes get attached themselves by the equatorial plate. Okay. Now each metaphasic chromosome is made up of 2 sister chromatids. That is very well known to you. As I have already told you, small disc-shaped structures called kinetochores, which are present by the sides of the centromere, are formed to which spindle fibers are attached. You know it. Now metaphasic chromosomes are arranged in such a way that 2 chromatids of each chromosome are attached to the spindle. Okay, 2 chromatids of each chromosome are attached to the spindle fibers of opposite poles through their kinetochores. Okay, they are arranged in such a way that the 2 chromatids of each chromosome are attached to the spindle fibers of opposite poles through their kinetochores. I will draw the diagram. Now metaphase is considerably shorter than the prophase but slightly longer than the anaphase. Remember, the most important thing during the metaphase stage is that the metaphasic chromosomes are arranged on the equatorial plate. Okay, by the time of metaphase, nuclear membrane and nucleus have totally got disintegrated. Got it? So the chromosomes are arranged at the equatorial plate in such a way that the two chromatids, you know that each chromosome is having two sister chromatids. The two chromatids are attached to the spindle fibers of opposite poles through their kinetochores. Okay, so how they are arranged? Let us see. Now, this is the diagram. I am drawing a big picture. Okay, now the metaphasic chromosomes are arranged at the equatorial. Equatorial plate means just the middle part. Okay, this is the equatorial plate. Got it. So how they are arranged? In this way. One, we had taken 3 chromosomes. 2. Okay, now these 3 chromosomes are arranged in the equatorial plate. How? In such a way that the opposite chromatids are, or 2 chromatids of the chromosome are attached to the opposite poles. Okay, this is how they are attached. Got it? This is how they are attached. Okay. Okay, this is how they are attached. Now let us come to the next one, that is anaphase. So what happens during anaphase? Now you have seen that all the metaphasic chromosomes are arranged at the equatorial plate and in such a way that the 2 sister chromatids are of each chromosome are attached to the spindle fibers of opposite poles. Now during the anaphase stage, the chromatids, as you know that chromatids are attached to the mitotic spindle, the microtubules pull sister chromatids in opposite direction. I have already told you the 2 sister chromatids of the metaphasic chromosomes are attached to the spindle fibers of opposite poles. Now these opposite pole spindle fibers, they pull the sister chromatids in opposite direction, as a result of which the centromere splits. Okay, the centromere splits into 2. Now the sister chromatids separate from each other. Obviously, as the centromere splits, the sister chromatids will get separated from each other, and then they move towards opposite poles. Okay, in such a way that the centromere faces the poles, whereas their arms trail behind. Okay, they face the equator. That means as the sister chromatids are pulled towards the opposite poles, the centromere faces the poles and their arms are directed towards the equator. Got it? Now this movement is due to repulsion between the centromere and the contraction of spindle fibers. Okay? Now anaphase is the shortest of all stages. Remember it? Anaphase is the shortest of all stages. Okay. So how this happens? Let us draw again. So as usual, this is the cell wall. Already nucleum and nucleolus has disintegrated. Now during this anaphasic stage, you have the chromosomes with splitted centromere. That means how they are pulled, just look at it. If this is the centromere and this is the 2 chromatids, now we had drawn 3, another one centromere. Okay, now another one, another 3. Okay, now this one. Okay, now as far as centromeres are concerned, these are directed towards opposite poles. As you can see here, this is the centromere, this one is the pole. So this centromere is directed towards opposite poles, whereas the 2 arms are directed towards the equator. Got it? So this is how the anaphasic movement occurs. Now let us go to the last one, that is your telophase. Well, as far as telophase is concerned, now by the beginning of the telophase, the chromosomes have reached the opposite poles. Okay, during the anaphase movement, the chromosomes were moving towards the opposite poles. Now, by the beginning of the telophase, the chromosomes have reached their respective poles. Now, after reaching their respective poles, they decondense, uncoil, and now they get lengthened. As a result of which they are not clearly seen. They are not as clear as the metaphasic chromosome. Got it? They are not as clear as the metaphasic chromosome. Why? Because after reaching the opposite poles, they have decondensed, uncoiled, lengthened, and thus not clearly visible. Now, at the same time, the spindle fibers which have pulled these chromosomes to the opposite poles, they have disintegrated. Okay, and another important thing that is formed during this stage is that nuclear membrane and the nuclear loss again reappear. Okay, so by the end of the telophase, the nuclear membrane and the nuclear loss again reappear. So what happens during telophase? Let us see. So as usual, this is the cell wall. Now chromosomes have reached the opposite poles and nuclear membrane and nuclear loss have formed in the opposite poles. This is the nucleus, this is the nucleus, inside which chromosomes are present. Okay, very thin and not easily visible. So now this one is the cell wall, this one is the nuclear loss. Just a second. Nuclear loss. This one is the nuclear membrane. Got it? So now let us come to the next one. After the telophase has been completed, that marks the end of the karyokinesis. So karyokinesis, what it includes? It includes the division of nucleus, which is completed in prophase, PRO-METAPHASE, then your metaphase, then anaphase, then telophase. So after telophase has been completed, karyokinesis has come to an end. Now the cytokinesis occurs, that is the division of cytoplasm. Okay, division of cytoplasm occurs during the cytokinesis. As far as cytokinesis is concerned, it is of two different types. In two different ways the cytokinesis occurs, that is the division of cytoplasm occurs. It is separate for the plants as well as the animals. In case of animal, the cytokinesis occurs by means of cell furrow method. Okay, as far as animals are concerned, the cytokinesis occurs through cell furrow method, whereas in case of plants, the cytokinesis occurs through cell plate method. Okay, as far as cell furrow method is concerned, here I have already told you it takes place in animal cells. What happens? Here, invagination of cell membrane from the equatorial region occurs. So here what happens? Already the invagination has started during the telophase. Okay, already the invagination has been initiated. Invagination of cell membrane has been initiated from the equatorial region during the telophase. Here further deepening of this furrow occurs, as a result of which two daughter cells are formed. So what happens during this stage? Let us see. Okay, let us see what happens. Here you have got what? Two nuclear membrane, nuclear loss, then chromosomes. So the cell membrane, the invagination has already started. Okay, as in this your cell furrow method, which is peculiar in case of animal cells, the invagination enters or deepens to such an extent that the total cells get separated from each other to form two daughter cells. Okay, this is the method of cell furrow method. Okay, now let us come to the cell plate formation. It is peculiar or it is typical of plant cells. What happens? The cell plate formation begins at the center of the cell and proceeds towards the periphery. Okay, it occurs from the center of the cell and proceeds towards the periphery. Now cell plate formation begins with Golgi derived vesicles which coalesce on phragmoplast that occurs on the equatorial plate. These phragmoplasts have been formed at the equatorial plate. Now they actually initiate the formation of cell plate. Cell plate represents the middle lamella between these walls of the two adjacent cells. I have already told you the cell wall structure: primary cell wall, secondary cell wall, middle lamella, tertiary wall. Got it? So cell plate actually represents the middle lamella between the walls of the two adjacent cells. As far as this new term phragmoplast is concerned, what is this phragmoplast? These are actually plant specific structure. Okay, these phragmoplasts are plant specific structures which forms during late cytokinesis. These phragmoplasts are formed during late cytokinesis. It actually acts as a platform for cell plate formation. Okay, these phragmoplasts are plant specific structures which actually get formed during the late cytokinesis and helps and acts as a platform for cell plate formation or cell plate assembly. Got it? Now, so this is how the cell plate formation occurs. So let us come to the significance of mitosis. What is the importance of mitosis? First one is, you know very well that it maintains genetic stability. Okay, because it results in increasing the number of cells. Even though number of cells are increased, from one cell two cells are formed, but the chromosome number remains same. So it maintains genetic stability. Another thing is that it helps in growth. You know, somatic cells or vegetative cells of all organisms divide mitotically and hence add to the growth. Got it? So the mitosis aids in growth. Got it? Next one is, it also helps in cell repair. Mitosis plays a very big role in repairing the damaged cells. So this is what all about your mitosis process. Okay, I hope this video will be beneficial for you for your future reference. Thank you very much.